Muted.
Hi, my name is Deirdre Mason, and I am with the Association of State Drinking Water Administrators. And today's webinar is being held in coordination with USGS on cyanotoxins, advancing drinking water science to protect human health. So for those of you who don't know about ASDWA, our mission is to protect public health and the economy through the provision of safe drinking water. Our members are the Drinking Water Program Administrators from the 50 states, territories, the Navajo Nation, and the District of Columbia, who provide public water system oversight in the form of regulations, technical assistance, and funding, and they also coordinate with multiple partners. This is the second webinar in our series with USGS. The first one was held on April 24th, and we'll have the other webinars ongoing throughout the rest of 2018 on these topics that you see here on the screen. So stay tuned for announcements about those coming up in the next few months. And for today's webinar, the purpose is to provide information about cyanotoxin occurrence and potential exposures in the nation's surface water resources, to share information about tools to quantify changing water quality conditions that may affect drinking water treatment processes and potential cyanotoxin exposures, and help the audience better understand and be prepared for cyanobacteria-related events. So our presenters today from USGS are Keith Loftin and Jennifer Graham, and we also have Ruth Briland from the Ohio EPA Division of Drinking and Groundwaters. Just a few housekeeping items. After the webinar, we will have a video recording available on ASDWA's website within the next few days or maybe week with the holiday coming up. And we ask you to please type your questions into the questions box. We are not going to be taking any verbal questions, and we will hold all of the questions till the end of the presentations. So at the end of the webinar, we'll be answering those. And with that, I'm going to go ahead and have Jennifer and Keith start off their presentation. So if you'll bear with us for just a moment. Jennifer and Keith, are you ready to go? All right.
We are, thank you.
Can you see our slides?
Yes, it is in presentation mode. I'm not sure if you can change the mode there.
Yep.
There we go. That's great. Thank you.
Well, thank you for the opportunity to talk today. We're going to be talking about cyanobacteria and cyanotoxins. This is a global issue, something that we've been talking about for a long time. One of the challenges with cyanobacteria is they can present very visual cues of their presence in the environment. And we really have no indication of whether or not that particular bloom is producing a toxin or not. So in this first title slide here, there's two examples of blooms, where one on the left is a bloom of cyanobacteria that has toxin concentrations in the hundreds of micrograms per liter, whereas the one on the right actually did not have any detectable levels of toxin. But just by looking at these two, there's no way to differentiate. And that continues to be an ongoing challenge when we're talking about cyanobacteria and cyanotoxins, is because of these visual cues, there's often a perception of risk to human health, but we don't know whether or not there's an actual risk until we can measure the toxins in the environment. So what Keith, Ruth, and I are going to talk to you about today we're going to give a little bit of background information on the cyanotoxins. We're going to talk about some of the approaches that USGS and the Ohio EPA have used to study cyanotoxins to better understand occurrence in the environment, risk to human health, and the development of tools to guide decision making. USGS will be focused on our approaches in science, and then Ruth will talk about the approaches that the Ohio EPA has taken and give some case studies. We'll talk about the prevalence of cyanotoxins in surface waters in the United States and potentials for exposures. And we'll also talk about technologies that may enhance source water monitoring programs if we can develop tools that give an early indicator that cyanotoxins may be present in the environment. Drinking water treatment strategies can be proactive rather than reactive in responding to an event after something is ongoing. So when we talk about cyanobacteria and cyanotoxins, we also we tie those in with harmful algal blooms. Harmful algal bloom is a term that we've all heard and one that's often used, but there's a lot of ambiguity and inconsistency in what harmful algal bloom means. When we're studying freshwaters, oftentimes we're thinking about cyanobacteria and cyanotoxins, but there are other organisms that produce toxins and cause concern in the environment. And so we need to be as specific as possible when we're communicating and talking about these issues. So today we're talking about cyanobacteria and the cyanotoxins. Something that we need to keep in mind and think about is what actually puts the H in HAB when we talk about algal blooms, what makes them harmful. Cyanobacteria and other organisms in the environment cause human ecosystem and economic concerns, but often that harm is not quantified. And often the harm can't be quantified until after an event has occurred. And so that's something we need to move towards more quantitative exploration of what we mean when we talk about harm. In addition, abundant algae or cyanobacteria aren't always inherently harmful. And the adverse impacts caused by them must be carefully defined. The term algae is one that often causes confusion when we're talking with the general public. Algae no longer has any kind of taxonomic meaning. What was once defined as microscopic plants is now known to include bacteria, plants, and animals. And as our genetic tools and probes have increased and improved, we understand that algae are a very diverse group of organisms. And so this is one, again, that causes confusion. And so we'll be talking about cyanobacteria today. As you all know, biological phenomena in surface waters can cause drinking water treatment challenges because of changes in water quality, because of metabolites and degradants that these biological phenomena can produce. When we're talking about cyanobacteria, often there's increases in biomass that can cause treatment concerns associated with clogging of filters. And the cyanobacteria also produce the cyanotoxins. Taste and odor compounds are another thing associated with cyanobacteria that cause drinking water treatment concerns, and we will touch on those very briefly today. And there's also concern with degradation products associated with the cyanotoxins and taste and odor compounds. All of these lead to treatment concerns and human health concerns. And often, all of these, the need to protect human health is balanced with the economic challenges, with the need to appropriately treat water sources, whether it's increasing our carbon feed, increasing oxidant, blending or switching sources, or providing alternate supplies.
So historically, if you look at the chart on the right, there's a variety of cyanotoxins, and this chart represents only a very few of the known toxins produced by cyanobacteria. Along the x-axis at the bottom are acute LD50s based on intraperitoneal injection in mice that have been reported in the literature. And on the right side, we have things that are toxic to us that have in the past elicited some level of concern, in many cases fear. Rattlesnake venom, chemical nerve agents, ricin, and the LD50s of the cyanotoxins as part of the bar chart, showing you where these fall in terms of these acute exposures. A range of effects have been identified with a variety of these toxins, both acute and chronically. However, there's much work that still needs to be done with human exposure testing. As you can imagine, there's very few volunteers and very few case studies where we can actually focus on human testing, which is why we have to rely upon testing of animals and other methods that we can go to get this information. One note that I'll mention that USGS is currently working on a critical review from a methodological basis on the toxicity information. Historically, these values have used a variety of different study approaches. One of the things that many of you already are familiar with from an analytical methods approach is we've just had a lack of certified reference materials. And several of these dose response studies used to derive the LD50s for this literature do not include measurement of the toxin. They trust what's on the label. And as many of you are aware, that's an issue for understanding and quantifying the amount of toxin that's present. And so we're trying to understand if this has a real impact on the setting of the LD50s and how those get promulgated into health advisories and regulations. So please stay tuned for that, and we'll see what the outcome is. Previously, in 2015, EPA put out a health advisory for microsystems and cylinders for spermopsin, and they went with a binary approach that's based on body weight. In reality, this is more like it'd be leveled at each weight, that you would want to compare it to for these toxicity values, but it's not reasonable to try to treat water to multiple points. It's probably a headache more than anything that you all could tell us a lot more about. So what the EPA did select to do is focus on the impacts of, being protective of bottle-fed infants, preschool kids, immunocompromised, pregnant mothers at the lower threshold of 0.3 micrograms per liter for microsystems, 0.7 for cylinders spermopsins, and then at a higher concentration in finished drinking water for school-age kids and adults, 1.6 and 3 micrograms per liter. If you'd like to read more information about the setting of these thresholds or some treatment guidance advice put out by EPA, you can see the 2 links below. One thing I will note with this is actually a 10-day average exposure, not just a single-day exposure. One of the challenges is knowing when the first day started if you're reactive versus if you can see the bloom coming into your plant and stay on track with what toxin concentration is present.
Okay, we know that cyanotoxins are widespread and common throughout the United States, and all states experience some kind of issues associated with them. So as an example, in May of this year, at least 15 states had recreational beach closures or health advisories associated with cyanotoxins in lakes and rivers. However, these are all based on anecdotal reports, and the information is collected in a variety of different ways, whether it's simply just a visual indicator that there's a cyanobacteria event or whether cyanotoxins have actually been measured. So this anecdotal type reporting really presents challenges when quantifying cyanotoxin occurrence. And then when we're talking about in finished waters, there's an added challenge there because there is no official way to collect and gather these kinds of information so that we can look at them in a scientific and quantitative manner. There are examples, several of which are listed here, where we have had cyanotoxin breakthrough in drinking water treatment plants, and that's something that information will continue to be gathered. But when we talk about occurrence in the environment and occurrence in source water supplies, that doesn't necessarily translate to breakthrough into drinking water treatment plants and then exposure and effects on human health. And as Keith mentioned, effects on human health, those kinds of data are very, very rare. The CDC did roll out a One Health harmful algal bloom reporting system, which is voluntary reporting for cyanobacteria and other algal-related events. But one of the things that USGS is working towards is trying to be as quantitative as possible when talking about cyanotoxins in all of the nation's surface water supplies, as well as those in source water supplies.
So from an occurrence perspective in potential source waters as well as waters that we recreate in, USGS is conducted in collaboration with US EPA, NOAA, the Army Corps of Engineers, and a variety of state agencies as well, to look at national occurrence and regional occurrence and potential exposure to cyanotoxins. Focused on lakes and reservoirs largely, but also wetlands. And you can see in the map on the right lower corner, those are the sites where we've seen a lot, we've done work already, and in a variety of surface water types. The last half of this slide continued. We've also been focused in estuaries, which has some relevance to there's some work being done on RO plants along coastal regions where a lot of our population is very dense, wadable streams, and then Jennifer's leading a study on rivers at a fixed site network. And so the main point we're going to talk to you today is that we've found cyanotoxins in all surface water types. The concentrations vary greatly, their persistence varies, etc. And so as we move into the future, this has relevance in terms of when do we, how do we select water supplies for various uses and so forth.
So USGS is consistently working to develop our field and laboratory methods to document those to ensure that data are collected consistently throughout the nation. And we're focused from field measurement and collection in the field as it moves through into the laboratory, and then as data are delivered and science is published. Those consistent guidelines are essential for capturing a national picture, and for cyanobacteria in particular, because of their ability to accumulate and cause those visual cues. It presents challenges to sampling and sampling consistently across and among sites. And so we have established guidelines for field sample collection that has been used on a worldwide basis. We also are working continually to improve laboratory analytical methods. Keith will touch on that. Morphological taxonomy, identifying the organisms that we're studying, is something that we're working on, and combining those with molecular methods to make sure we're calling things by the right names. And there's other approaches that we're developing that we'll touch on briefly as well.
So among the many challenges we deal with on this issue is the fact that there are a wide variety of classes of toxins, as well as number of toxins within those classes. They don't all share the same mode of action from a toxicity standpoint. And so we've been using converging lines of evidence approaches to try to better understand and grapple with this issue to understand when a water truly is safe. So across the top, we have in gray the cyanotoxins that we all typically are focused on, with most of the emphasis being on microcystins, but it includes anatoxins, cylindrospermopsins, microcystins. Nodularins not as much in freshwater, but we do see it occasionally, mostly in elevated salinity circumstances though. And then saxitoxins we're finding. However, there are a wide range of known compounds that have potential effects that need further research, understanding what their true impacts are. And it just further underlines the mixture potential that we see and have to deal with on this issue. And that includes aeruginosins, aminopeptins, anatoxin AS, biogenic amines. Usually that's associated with food poisoning with fish and shellfish, but they are available in cyanobacteria. Cyanopeptylins, the depsipeptides, lipopolysaccharides. We've done a little bit of work with lean biotoxins, but there's a shortage of analytical standards for those, and so on. So, and this doesn't touch on everything, it's just what we know right now. And so I would say very clearly the ones in white are understudied for sure. Along the left-hand side are some of the known modes of action. So it doesn't mean that a toxin class only has a single mode of action, but it's the primary mode that's been studied. And the circles just mean that USGS currently we have capability to do a mode of action assay in our laboratory, and these are commercially available assays that we compare with the chemistry we do with quantitative and then screening with ELISA to make sure that we can understand what the potential mode of action is, potential effect, if you will, compared to the chemistry. And we need both to move forward, trying to mesh with the One Health concept. The other thing the mode of action assays do for us is it allows us to know if there are other chemicals that may be present that causes a similar effect to the cyanotoxins that are present. And so moving forward, we're working to describe that more explicitly. Within our laboratory in Kansas, we have both targeted and non-targeted screening and quantitative analysis. We use a range of 96 well plate approaches for the bioassays, and mass spectrometry and spectroscopy and so forth is needed depending upon what the research question is. And then those methods end up getting transitioned into monitoring type approaches to help us get a better idea of what's going on in the nation. Or we're also able to drill down more specifically for case studies. In our lab specifically, we've been focused on protein phosphatase inhibition, acetylcholinesterase inhibition, and nicotinamide acetylcholine receptor binding. And then for the targeted screening with ELISAs, there's now an anabinopeptin ELISA that we're working on validating. And then anatoxins, cylinders, spermopsins, microcystins, nodularins, and saxitoxins, which you all are more familiar with probably. And then from a triple quadrupole mass spectrometry approach, we've also got counterparts for the first 4 classes of toxins, and we're working on one for saxitoxins as well. We do see saxitoxins in freshwater, and we'll talk with you about that in a moment. So as part of our effort in USGS to better understand human exposure as well as ecological exposure in this issue, we want to keep in mind that human exposure is really it's a complex equation and focused on recreational drinking water and food exposure. And it comes from a variety of sources, etc. And from an analytical method standpoint, that can be a nightmare for trying to validate a method in each matrix, because that's a new method to validate, for each organism, if you will, because the tissues are quite different chemically in a cow versus a fish, let's say. And so we have to prove that the methods work in all of those cases. The other thing is we want to know that just because a contaminant is present, it doesn't mean that there's an implied harm that's going to be caused. And so one of the things we're working on is developing bioaccessibility extraction methods with simulated saliva, lung fluid, gastric fluid, and intestinal fluid to see if we can extract toxins or other contaminants that might be related into a gastric fluid, for example, and does that make it into the bloodstream? And then we can form the metabolites and use our non-target screening approaches with accurate mass spectrometry to measure biomarkers and work with people on developing biomarker assays. And hopefully what we can do then is help triage and prioritize which issues we need to be focused on the most, and whether recreation, drinking water, or food needs to be our larger priority, given those circumstances. And so we plan on taking finished drinking waters that we spike with toxins, as well as look at fish and other food sources in this process as we develop and validate these approaches to see what toxins make it out and what biomarkers are present.
So some of the other ongoing work that USGS is doing that you'll need to stay tuned for results is looking at the environmental effects of exposure on aquatic organisms. And as Keith mentioned, we're developing the tools to look at biomarkers, and perhaps those biomarkers in aquatic organisms could be used as sentinels to indicate potential for human health risk or human health exposure. Some of the ongoing work that we have, this is just one example, working with the cyanotoxin microcystin in endangered fish in Klamath Lake, and preliminary and early results from this study indicate that microcystin may affect recruitment of juveniles in this system. And again, you'll need to stay tuned for that work as it progresses. Right, some of the other things that we're doing that aren't focused on direct measurement of cyanobacteria and cyanotoxins are near real-time data acquisition and delivery through a variety of different types of platform to give that early indicator that something may be going on that may cause human health concern. We're working at a variety of scales, from satellites down to sensors placed in the water and cameras. We'll give a couple examples of studies using satellites and sensors. We're not going to talk too much about cameras, but there are times when a picture is worth 1,000 words when we're wanting to just know whether we need to pay attention or be concerned as an intake or a specific site. So USGS and others that are studying cyanobacteria and cyanotoxins are really using converging lines of evidence across a range of scales, from what's happening at the subcellular and molecular level all the way up to satellites and Earth observation systems. And all of these tools are needed to move our science forward as we work to better understand occurrence, human health risks, and tease apart what the perceived risks are as opposed to the actual risks.
So some of you have probably seen the next few slides, so I won't stay long on them. But in 2007, we did a national lake survey with EPA for microcystins, and then afterwards we went back and looked at cylindrospermopsins and saxitoxins. One of the key takeaways from this is that we did find toxins nationally, not just locally, which had been the prevailing thought of some folks, but it is a global issue to pay attention to. And we see that, in this case, we weren't looking at the worst case scenario, but out in the middle of the lake at the surface, instead of near shore like we do for recreation a lot of times. But we still found 32% of the samples across the country, over 1,000 lakes, had detectable levels of microcystin. Most of them weren't elevated according to the WHO thresholds, but they were there. Likewise, you'll find that the patterns between the different classes, the last slide being microcystins, this slide cylindrospermopsins, can be quite different. And the main takeaway for this slide and the following slide is just because you measure for microcystins doesn't mean that you're going to know what the situation is for other cyanotoxins. And so it's something that we have to pay attention to. And I think I've sent that message pretty clearly to folks in the past. We had about 4% cylindrospermopsin detections in this study and 8% saxitoxins in this study.
And as Keith mentioned, just because you measure microcystin doesn't mean other toxins aren't also there. And in this particular study, we were looking at visual accumulations of cyanobacteria, but testing for multiple compounds. And in this study, we found that multiple compounds occurred simultaneously, including multiple classes of cyanotoxins, as well as taste and odor compounds. And while that doesn't necessarily mean that they're always going to occur together, or one can be used as an indicator of another, it does indicate that oftentimes when we're talking about drinking water treatment, we may be dealing with complex mixtures. And if you're optimizing for taste and odor removal, that does not necessarily mean that you're also optimizing for cyanotoxins. And so that's something to keep in mind, is that we're dealing with complex mixtures of different types of compounds produced by the cyanobacteria.
So another series of studies in wadable streams. This particular study is in Southeast United States, down in the lower right-hand corner from 2014. But we're doing these wadable stream surveys to understand if the wadable streams might be a potential algal loading or cyanobacteria loading, to be more specific, scenario for the cyanobacteria itself or the toxin into receiving waters like lakes and reservoirs. And we did find microcystins present. None of them were above any recreational threshold of concern, but 39% of the streams in the Southeast had detectable levels of microcystin. Again, it doesn't mean it's an issue. We don't know fully what the potential ecological impacts from this being present could be, as well as the benefits. And so that's something we're working on as well. We have other studies from across the United States that we're wrapping up and we'll be publishing probably within the next year.
So in a lot of our efforts on cyanobacteria and cyanotoxins have been focused in lakes and reservoirs. However, we have seen in the news in recent years, starting with the Ohio River, that cyanobacteria blooms can develop in large rivers as well, and we're seeing this summer development on the Willamette River in Oregon as well as the Illinois River. And so one of the studies that we're working on now is to look at cyanotoxin occurrence in large river systems. And this is some preliminary data that was collected last summer from 11 large river sites across the nation. We did detect the cyanotoxin microcystin at 2 of these sites indicated by the stars. We did not detect any other cyanotoxins in this study. However, we did detect the genetic potential for cyanotoxin across all sites except 1. And so that indicates there is potential for occurrence at these sites if conditions were to change. What that genetic potential actually means and how conditions may change to favor that is an active area of ongoing science, and Ruth will touch more on genetic tools and genetic potential and how they may be used. Another challenge when working with large rivers is understanding whether the cyanotoxins that we're seeing are being produced in the rivers themselves or being transported from upstream lakes and reservoirs. We do know and have documented that cyanobacteria, cyanotoxins, and taste and odor compounds may be transported for relatively long distances downstream from lakes and reservoirs. This is just one example where there was a large bloom on an upstream reservoir that had documented human illnesses and hospitalizations as well as dog deaths. And water was released from the reservoir during that event, was transported over 180 miles downstream, affecting multiple drinking water intakes along that study reach. The study also indicated here that there is some measure of ongoing growth once the cyanobacteria reach the river. And again, those are science questions that we're still looking to address, is when is transport occurring and what is the state.
So in 2017, USGS conducted a tap water exposure study for not just cyanotoxins, but for a range of contaminants and biological targets of concern. And I'm going to only talk about cyanotoxins because the rest of this will be covered at a later talk, I believe in August. But we had scientists that actually went into homes of volunteer people in the greater Chicago area. There were a variety of water treatment plants represented. One of the largest plants in the world, 1.5 billion gallons per day, was one of the supplies. And then there was also a variety of smaller supplies. Most of this water was coming off of Lake Michigan for that area, but there was also some other sources. But to date, we have not had a single detection of cyanotoxin in the finished drinking water from this reconnaissance. And so, you know, this is just part of how we're trying to get a better understanding of when these issues need to be dealt with, and this is ongoing work. And you'll get more information on that later in August. So another aspect that we're working on, point of use drinking water treatment has become more of a in vogue way of dealing with water. And part of it is in concern with water quality, even though we have a high quality of water coming from our nation, and then accessibility as well. But then the other aspects are when people are in remote destinations recreating, you know, the personal backpack drinking water filters and things like that. A lot of claims are made, but we don't always have the science behind it to understand if it really holds up when in real use. And so one of the things that we're working on this year, developing the methods and approaches to test, you know, some of these treatment options. We've done some preliminary work on this in Alaska, where we had a source water that our field crew was drinking from. And we had around 60 part per billion of microcystin. And they were using a cheesecloth and filtering out the larger algae, but then they were drinking the water because they were on a remote trip, of course. And they did find some breakthrough in the backpack filters they were using, but it wasn't above 1 part per billion, which was the WHO guidance at that time. And so we're working on seeing what treatment options are available to the public, and seeing what really is the issue from this scenario. The other application is when we get do not drink orders, or we have extreme events like the hurricane down in Puerto Rico. We end up having to ship a lot of bottled water or things like that. If people could use these types of technologies, and if they're easy to use and so forth, that might be something where we could be a little more proactive on the rare occasion where we have to deal with these types of issues. So you also may be aware that USGS, in collaboration with NOAA, EPA, and NASA, have been working on detecting cyanobacteria blooms from space using satellites. And to date, we have national coverage for the continental U.S. For larger lakes that we can see with the Sentinel-3 satellite, and then we've done some historical work with Maris and MODIS, and we're trying to make some headway with Landsat and Sentinel-2 platforms. Some of the states on this call are already collaborating as stakeholders on the project. Ohio, who will speak later, are one of those state stakeholders. But the main idea here is to identify these blooms as they start to grow and potentially hit drinking water sources, excuse me, as well as recreational situations, and get a better national, more consistent review of how this issue is progressing, because people make claims about if the blooms are getting worse or not, but we don't really have the consistently collected data sets to know that. And point samples aren't the way to always understand that issue in the first place. One of the things that we're trying to do is integrate ground, laboratory and field data with the satellite approaches as well to reinforce the ruggedness of this approach. It's not a perfect approach, cloud cover being one of the biggest issues. The satellite can't see through them currently, but you're getting more information than we were able to before. And so one of the things that EPA has done a nice job of is making a mobile application where you can input your thresholds. And this is in beta testing mode right now, where based upon the satellite imagery that's being collected, it can give you a feedback in terms of what your risk threshold may be for cyanotoxins. And so this should give you maximum flexibility to put in your own thresholds if you're more restrictive in your source waters. And it allows people to have information in their hands to make decisions about where they recreate, and also what may be going on in their source waters.
One of the challenges with satellites is the time scale, where we're looking at a few days to a few weeks overpass. And water quality sensors that have the ability to take measurements at much finer time scales have shown promise as early indicators of developing events, which could inform monitoring strategies, could inform treatment processes. The challenge with the water quality sensors is that they are at fixed site locations, and so they're representing a single location. There's a lot of different ways that the data from these kinds of sensors can be used as an indicator for the presence of cyanobacteria or cyanotoxins. And one is simply looking at the raw data streams. The examples here, the one on the upper left is simply looking at pH in green, dissolved oxygen in blue, and those wide swings over a 24-hour period are indicative of biological activity. And so that may be an early indicator that there is biological activity, and you may want to increase your monitoring or measurement for cyanobacteria or cyanotoxins. Another one on the lower right is based out of a fluorescence sensor that is tuned to the cyanobacteria. And then looking at that, we call this the cyanobacteria seismograph, where an increase in the noisy signal may be an indicator that there is cyanobacteria present, and again, may serve as a warning or an indicator to increase cyanotoxin testing. Because of the patchy nature of cyanobacteria bloom development, this kind of seismograph is really just indicative that there may be patches of cyanobacteria moving across the front of the sensor as depicted in the photograph on the upper right. The other thing that we can do with these types of sensors is develop models to compute the probability of cyanotoxin occurrence. So while there really aren't cost-effective sensors out there yet to serve as a real-time indicator of cyanotoxin presence or concentration, we can develop models that would serve as an indicator. And so the example shown here is a cyanotoxin model. It's estimating the probability of microcystin occurrence above 0.1 microgram per liter in the drinking water supply for the city of Wichita. So the black line is the estimated probability of exceedance, and then the red there is the actual measured microcystin concentrations. These kinds of models have been developed by USGS at several sites in Kansas and Ohio that are operational, and that type of information is being served on the web through a variety of platforms, be it just a continuous estimate or 15-minute estimate of the probability of exceedance as shown in the upper right, or whether it's more of an indicator on a more of a stoplight, no issue potential problem or problem scale as shown on the lower right. One of the challenges with these types of models is that they are site specific and they may change depending on changing conditions. This is an example from Cheney Reservoir in Kansas, where we have studied cyanobacteria and cyanotoxins for almost 20 years and felt that we had a good understanding of the causes of cyanobacteria and the compounds and when we would see them. And then in the summer of 2013, we had the largest single inflow event over the course of the study, and it really reset the system. In this system, we often saw small winter peaks in the taste and odor compound geosmin and summer peaks of microcystin. What happened in 2013 was after an extended period of drought, we had one inflow event followed by a larger inflow event that led to summer geosmin episodes. And this new pattern has stayed and remained. So this is just to demonstrate that just because we think we have a system figured out doesn't mean that conditions might not change and lead to completely different processes and recalibration of models. The other thing that we can do and have been able to do in some instances, this is another example from a Kansas reservoir, is that we can use the water quality monitors to actually develop models to compute cyanotoxin concentrations. So we were able to develop a model to estimate microcystin concentrations on an approximately 15-minute basis. And again, it's another indicator of what's going on in the environment. This particular example is actually from a recreational water supply and is showing estimated microcystin concentration in comparison to the advisory levels that the state uses to post recreational warnings. I wanted to include this as an example because, again, it demonstrates the variability in microcystin concentrations at a single site over time. As you can see, it can change very rapidly, and that does have potential implications when you're looking at trying to monitor to protect public health, and all of those things need to be taken into consideration. And so in trying to use this type of information, we're still working on approaches and best practices, but they certainly do show promise.
So as you can see, what we've been working on for the last decade and a half and where we're going into the future, we're still focused to some extent on status and trends in fate and transport drivers for these issues, but we're also moving more into exposure and health as well as the ecosystem effects to understand the real versus perceived risks. Trying to differentiate the drinking water and food impacts, we think is going to be critical to defining, where this issue really is an issue and moving forward. And then ultimately, when it's necessary, how do we go about mitigating and managing these problems? And so USGS is going to continue to use converging lines of evidence to have rugged science to help define these issues and try to fill in the data gaps that remain.
Great, thank you so much, Keith and Jennifer. We really appreciate your presentation. So next we're going to be moving on here to Ruth from Ohio. And I just want to remind everybody to please type in your questions in the webinar control panel in the questions box, because we'll be taking all of the questions at the end after the presenters are finished. So Ruth, I can see your presentation. If you could maybe push F5 to launch your slideshow. There we go, great. And I think you need to, oh, there we go.
Unmute, yes. Very good. All right. So a swift transition into Ohio as a case study. And I really feel like looking at our timeline of Ohio's response to harmful algal blooms is the best way to kind of organize this presentation. As you see, our response really just began only 8 years ago in 2010, which was basically incident response sampling due to observations. And within a year's time, recognized that we do have some, the harmful algal blooms are an issue for the state, and they brought together our primary agencies with Ohio EPA, responsible for drinking water, Ohio Department of Natural Resources, who's responsible for recreation and manages many of the water bodies, and of course, the Ohio Department of Health. And collectively, they created Ohio's harmful algal bloom response strategy, and at the time, it was integrated between drinking water for our public water systems and with recreation. This is when cyanotoxin thresholds were first established for our state. We had specified sampling approach and advisory language, and of course how to sample for cyanotoxins. And I want to point out that at that time, we used phytoplankton identification as our screen for other cyanotoxins. Of course, just a few years later, we had drinking water advisories for microsystems. The first actually occurred in 2013, and this was actually a small Lake Erie public water system where the advisory was issued, and it did not receive much media attention because the system was able to quickly transition to an emergency connection with a neighboring public water system, although it was not able to return to their source water for over a week until the microsystem levels in the source water had decreased. So this is an event that didn't get as much attention as the following year, when in 2014, this time out of a very large water system in Lake Erie, Toledo, we had a do not drink advisory was issued, and water had to be trucked in from neighboring areas, and of course the press coverage was international even, and really called for state and federal legislators to take action, which within the state of Ohio, they did. And US EPA released their health advisory levels for microcystin and splinter rhamnopsin that Keith touched on earlier, and that was integrated into the public water system and recreational strategies for that year. We had numerous outreach and engagement sessions, and also had finished water detections at 5 different water systems that year, but all had repeat samples that were below the newly established thresholds, and fortunately no advisories were issued. So it's a really good point to make that with the updated health advisory levels and taking into account that average over a 10-day window really allows us to incorporate that into our monitoring program and respond appropriately by optimizing treatment and reducing those finished water detection levels and avoiding drinking water advisories. So also in that year, in 2015, Ohio passed the Senate Bill 1 and led to Ohio's development of the harmful algal bloom monitoring and reporting rules, which I'll touch on. And then in 2016, those rules became effective in June, and we actually tested qPCR as a screening method, and I'll talk more about that in detail. And that Ohio EPA covered all the costs for the qPCR sampling and analysis for 18 months from June of 2016 through December of 2017 to evaluate that tool. And so in 2017, we were able to look at the current and treatment capabilities of the water systems and offer reduced monitoring or a tiered approach. And now we have finally shifted into full implementation of the HAB monitoring by the public water systems themselves. So it's a brief overview of the rule, and I won't go into too much detail here. You can look all of this up through Ohio Revised Code, but it does codify the microsystem action levels in rule and sets the monitoring and treatment technique requirements, as well as, of course, record keeping and then public notification. Of course, we need laboratories to analyze these methods, so that required revisions to a new Ohio Administrative Code, Rule 3745-90-4, and amending some other rules for lab certification and analytical techniques, as well as reporting deadlines. Now, how that all gets implemented is in a more general way read through our public water system HAB response strategy, and that's been updated every year since 2011, and you can find the most current version on our website now. And of course, it goes into more detail about the monitoring program using QPSTAR as a screen. The newer reduced monitoring options are in our current 2018 strategy, as well as our threshold response to finished water exceedances. So the numbers, as Keith and Jennifer mentioned earlier, we have in our harmful algal bloom strategy, we have 4 cyanotoxins identified with tiered threshold levels versus the do not drink for sensitive populations and children under the age of 6, a higher level of do not drink for including all populations, and then our do not use is really informed by our recreational level. Again, we're highlighting that for microsystems, the action level is actually in rule, and for all the other cyanotoxins, they are just listed in our public water system strategy. So as new information becomes available, we can update that in those cases. And then to highlight again for microsystems, it's based on total microsystems in that 10-day window. And we'll also highlight that any exceedances in the finished water will trigger additional monitoring, treatment optimization, and potentially other actions as needed. So in the rule monitoring requirements, we define the distinction between monitoring for total microsystems using ELISA, and this happens weekly, May through October. That's kind of known as our HAB season. And then we offer reduced monitoring on a biweekly schedule in November through April, with triggers that if any detection occurs in the raw water, then you're brought up again to the kind of in-rule sampling. And of course, not all of our communities receive water from within the state of Ohio, so any purchased surface water systems from outside of the state require finished microsystems monitoring year-round. And then the screen is using qPCR. Again, this is biweekly raw water only, and it triggers follow-up sampling by Ohio EPA for all the other cyanotoxins. The water system is only responsible for monitoring for microsystems. So what is our response? When we have cases of increased level in the raw water, the monitoring from weekly increases to 3 days per week as doing raw and finished. Any finished water detection, and this is not an action level exceedance, but any finished water detection would increase to daily monitoring. And of course, the Ohio EPA would immediately be in contact with the water system and hope to optimize and avoid an action level exceedance. However, in the case if that occurred, we have a monitoring program in place where you would resample within 24 hours of being notified and immediate analysis within 24 hours of collection, so that we have more evidence that this was an actual exceedance, and then repeat that analysis within 24 hours. All of this, if analysis occurs rapidly and there's good timing and communication, can happen within the 10-day window, because we'll have data, from the previous week demonstrating that there was no microsystems in the finished water. In the event that microsystems are detected in the raw water or finished water, there's 2 different treatment technique requirements that are triggered. One, our treatment optimization protocol is seen as a short-term or optimization utilizing the existing treatment capacity within the plant. And this is triggered by any microsystem detection in the raw or finished water. And at this time, we have over half of our water systems, actually 66 water systems out of 118, have had microsystem detections and triggered treatment optimization protocol. In the event where microsystems are detected on either the finished water or in the raw water greater than 1.6 micrograms per liter, and this is twice within one given year, so we're really identifying systems that have high probability or high occurrence in microcystin and not just individual blooms. And right now we have 19 water systems that have triggered this general plan. And it's much more involved, more holistic assessment of treatment effectiveness and needs, includes our source water protection activities, as well as reservoir management and implant treatment, and can include upgrades. So we do have guidance available on our website if and when these are triggered. I'll also add that all of our cyanotoxin data from our public water systems, as well as our Division of Surface Water, Inland Lakes Monitoring, and other Lake Erie sources, is available through our website. And so we've got a map portal here where you can see where all the sites are located and get a sense of where observations are currently happening. You can toggle between the different toxins or time periods, and so just note the type of sampling, if it's raw water or finished water. And then, of course, you can download all of the data directly through our webpage and look for the algal toxin results tab. So what do occurrences look like over time? And here we've just plotted any detection greater than 1.6 in Ohio. And we also have kind of listed what trends were like from year to year. And I think the good point to take home is that our highlighted period of harmful algal bloom occurrence, or bloom season, during the May through November, holds true. That's when the greatest occurrence happens in the late summer, especially with high microcystin detections. But we have had detections across all months of the year. So it's something to keep in mind, that having at least some type of year-round monitoring is helpful and informative. And in some cases, some water systems have only had wintertime issues, and in fact, not had microcystins detected in the summer. If we look at occurrence throughout the state using just our public water system data, here we've got overlaid with the blue dots is our surface water systems, black squares where saxitoxins have occurred. And in this case, I want to note for saxitoxins that toxin is only tested if and when the genes were present, and then that triggers follow-up monitoring for saxitoxins. But microsystems are tested independently. To delve into our cyanobacteria screening a little bit more, our molecular methods, we've given the detail here and highlighted the 4 types of information that we receive. So one is a gene specific to all cyanobacteria, or the 16F, and we'll refer to that as total cyanobacteria in some upcoming slides. The MYCE gene that represents microcystin or nodular production. In the case of these systems, we're really looking at it just as a screen for microcystins. CYRA for cylindrous, spermopsin, and then SXTA is the saxitoxin producing gene. The benefits of this is that it's a fast screen that can be done within hours. It is scalable, cost-effective relative to other options, and utilizes a certified reference material, and that's really key. And it's specific. If there is no gene, then we don't expect to see the toxin. In the case of phytoplankton ID, in some cases, you can identify a type of cyanobacteria that may be associated with those cyanotoxins, and therefore it's not terribly specific. There's a reference for the type of assay that's currently been in use. So how did qPCR serve as a screening tool for microcystins? And we were able to use the period where we had paired samples for microcystin and qPCR. 45 of our water systems had microcystins detected, and MCYE gene was detected at more than those 57% of those 57 water systems. And in the paired samples from 2016, all of the microsystems that had high levels, either greater than 1.6, we had paired MCYE genes at the time. And in many cases, the genes actually occurred just a week, maybe 2 weeks prior to when we would see microsystems. And another kind of level that we observed is that 100% of the microsystems that were greater than 5, which would trigger our 3 times per week sampling, had MCYE genes greater than 5. So we really started to look at this potentially as a screen for those high-level microsystems. And only less than 2% of our samples had the low-level microsystem detections without MCYE detections. So we're maybe missing very low levels of genes, and perhaps there is something that can be done to improve the lower end of the detection limit for qPCR. So here's an example. We look at time series trends with microcystin, the toxin presented in red with the MCYE gene below it, and the 16S gene, so representing our kind of total bloom size or total cyanobacteria. And you see that we see a strong increase in total cyanobacteria, and that's at a time when the MCYE gene is first detected and actually at its highest level, and then toxin production follows within 2 weeks. So here we think that's a great example of how qPCR performs as a screening tool for microcystins. And in a different case study, looking at Tappan Lake, where we have a longer history of microcystin data, and this is a system with a known microcystin issues, but the toxins occurred variable in the concentration and the seasonality of it. So in 2016, we had a peak during early summer and then in the fall to early winter. But in 2017, the peak actually occurred, toxin concentrations increased throughout the summer and peaked towards late fall. And when you look at that trend related to cyanobacteria screening data, it really shows how maybe there's something missing here. So in 2016, total cyanobacteria did not correlate well at all with the MCYE gene. So the gene was only present early and then late in the summer when total cyanobacteria was actually only at moderate levels. And in 2017, the 16S and MCYE followed very close trends that were associated with microcystins. And in this case, we really would like to look at more information to see did we have a composition change during these 2 years, or were nutrients an important driver, especially knowing that nitrogen is an important component to the microcystin molecule. And this is a great opportunity to mention that Donna Francie, a collaborator with USGS's Water Science Center, is actually delving into this issue and working more closely with this water system at exactly those things. So she's using a suite of different qPCR assays that look at the MCYE gene associated with specific types of cyanobacteria, like Planktothrix, and also collecting additional information for nutrients and looking at correlations between those. So we've seen some of her preliminary data and look forward to the final outcome of that study to explain this issue. And finally, this is just showing the data that we had talked about before, where at high cases where we have high microsystems, the red dots, we're kind of looking at the relationship between microcystin producing cyanobacteria using count data, and we did translate it to be on the same scale. So in cases where we have basically phytoplankton ID saying that yes, you have bloom levels, cyanobacteria that are capable of producing microcystin, they were associated with periods where we had higher MCYE genes and high microcystins. And if we look at the area in that dotted portion of the plot on the very low end, kind of blow it up on the left side, and that threshold with the dotted line, the vertical line at 100 cells per microliter or 100,000 cells per mil, which is our threshold for looking at a bloom. In this case, I think it also shows that only low to moderate levels of microcystin are found on that lower end. And so in this case, maybe utilizing phytoplankton ID would be a good screen for those higher levels, but not necessarily at the lower levels, because we still had low and moderate levels of microcystin present when in 2 events at least where the cyanobacteria responsible that we would associate with the microcystins were not present. And looking at similar plots for saxitoxin, we actually see a different picture. So when phytoplankton ID said that saxitoxin producing cyanobacteria were at bloom levels, all those X's along the x-axis actually, we did not detect saxitoxin. However, at very low levels, we see the saxitoxin or SXTA gene was present, and saxitoxins were also detected on that low end. So in this case, phytoplankton ID would not be a good screening tool for saxitoxins. And to drive that point home a little bit, we looked more closely at those data for saxitoxins. And here in the bar chart, we actually have the cells per microliter of the different genera of cyanobacteria that are associated with saxitoxin production, the SXTA saxitoxin production gene, and the saxitoxin levels. So only in 2 cases could we maybe consider that Cyanotoxins phomopsis or afanosomnon were responsible for the cyanotoxin production. But in other cases, especially the site D, we had 4 locations where only a tiny bit of Delcospermum or Andobena was detected by phyto ID, but we were still detecting similar levels of saxitoxin and the SXTA gene as other sites and places where much higher levels of cyanobacteria were associated with that toxin. So again, just highlighting that maybe phytoplankton ID is not the best screening tool for saxitoxins. Finally, to further drive home how well is our total cyanobacteria production or 16S gene performing, we did do an interlab comparison with US EPA, and we've got our R-squared line here, and it's a very strong relationship where the total 16S is performing well and utilizing the phytoxygen assay. So finally, looking at all of that and kind of considering our molecular methods and potential next steps, from our perspective, we really see that the qPCR is a good tool and that we're going to move forward with it. Refining, we still see there's opportunity to improve and refine sampling and analytical methods to better address the high biomass samples like scums and benthic samples. We basically, when we screen for this, we consider it more of a present absence than a true quantitative result. And now we are working, since this is a tool that our water systems are expected to use for analysis, we need more support from external labs, and so a lot of certification is ongoing. We currently have 4 vendors. From the US EPA partner side, they are assisting with method validation, doing interlab comparison, and sequencing samples for 16S to maybe learn more information about what cyanobacteria genera are present. And with other research collaborators, they're focusing in on the metagenomics side to identify the sources of saxitoxins and Cyanotoxins phomopsis that may, and what may trigger the toxin production. And altogether, we are responsible for not just generating data, but actually summarizing it in a cohesive way and getting it published that can help inform national policy. This is just a second summary slide that I think states it more clearly that we feel that based on these data, MCYE is an effective, specific, and good screening tool for microsystems. And our support for this is that any MCYE detection may indicate an onset of a bloom and could provide early warning. More severe blooms where we have those higher gene levels were always associated with higher MCYE levels. And we've helped build that relationship into our reduced monitoring schedules, where we have weekly data coming in from either the qPCR screen or microsystems, and we're utilizing the MCYE gene greater than 5 as a trigger for immediate microsystem analysis. And that saxitoxin A is similarly an effective, specific, and good screen for saxitoxins. We also, at some cases, got to see the demonstration of the multiplex functionality of the assay working well. We had one system where MCYE gene, saxitoxin A gene, and Cyanotoxins phomopsis genes were all present in the same assay. And that 16S may help as an effective screen for cyanotoxins. It's not an effective screen for the toxins themselves, but it is a potentially useful tool for assessing susceptibility and may help us with the in-system. And finally, phytoplankton enumeration is really not seen as an effective screen for saxitoxins, although it may be useful for screening microsystems at higher levels associated with our recreational thresholds. And I did want to touch on monitoring, perhaps utilizing cell light imagery. As Keith said, the state of Ohio is one of the partners and state-level stakeholders for the Cyan program. And so we've been given access, provided access with some of the cyanobacteria data products And we're just highlighting here that we generally get 3 products. One is total cyanobacteria index, and one is specific to what's associated with the cyanobacteria, and one is for other types of algae. And then we download this file essentially every day that we have data for, and then generate images or small maps for all of our inland lakes. And here's an example of Tappan Lake that I showed data earlier. And we've got our public water system intake indicated at that location. And so this is a map showing that the cyanobacteria is concentrated up, actually downstream near the dam, which is on the left-hand side here, but still near the intake. And so we share this type of information with our agency partners as well as the water systems and the, in this case, the entity responsible for monitoring that water body. And it has helped helped with oh yes, they have recreational sites at this water system as well, and it just helps guide sampling efforts and further supported the other data that we were seeing, that the water body has ongoing cyanobacteria issues. And with that, I think our slides are concluded, and I'll pass the baton.
Yes, thank you very, very much, Ruth. And so I just want to remind everybody to please go ahead and type your questions into the question box on your webinar control panel. And we do have a number of them that have already come in. And so the first question that we had was asked during Jennifer and Keith's presentation. And Jennifer and Keith, I want to make sure you unmute yourselves so you can answer the questions. It had to do with the harm in harmful algal blooms. And we have a question here about whether there are any confirmed chronic exposure cases in the U.S. But maybe Ruth, you could chime in on this one also in terms of whether you've seen any of that in Ohio.
Yeah, so Keith and I aren't aware of any kind of chronic exposure information from the United States.
Yeah.
Acute, there are acute examples, but not chronic.
The chronic case that I'm aware of, I don't think was documented well or at all. There was a case down in the south central U.S. where a lady had already had liver impairment and was swimming, but it wasn't a drinking water aspect. And then the study that was published on liver cancer, I don't think that fully captured all the variability and the issues that we need to deal with, because there are a lot of aspects covered in liver function, and I don't think that study really captured all of that. And in all fairness, our reporting systems don't really allow us to record all that information as well.
Okay, great. Thanks. Ruth, I don't know if you wanted to add anything to that.
No, unfortunately, I don't have any information to add on that, but I do think what I will note is it's important for I think the public to understand that difference, that our health advisory level is focused on that short-term exposure. And so if we start looking, just like Keith said, at the ways that individuals are cyanotoxins or microcystins through both recreation, eating a fish, and swimming, that's something that we're going to come up against, I think, more questions from the public on.
So along the same lines, and this question is for you, Ruth, is, how do you implement the 10-day health advisory action level? Do you, I know you mentioned that you require repeat sampling, but when do you provide public notice? When do you ask water systems to provide public notice if you have, the 10-day health advisory level? So how do you interpret that and implement it?
Okay, so I'll refer to our public water system strategy here so that you can read all the details. But in general, if when a sample is collected for microsystems, it has to be analyzed within 5 days. So you should have a result if there is a detection in that finished water sample within the first 5 days, which would allow for within the 10-day window the repeat and the resample to occur. So, and that's essentially when we do get that result of a finished water exceedance of the action level, we would look back to what was the most recent data leading up to that, and that would really start the clock ticking in a sense for what the 10-day window is. So it's really key here to get the results of any microsystem detection back to the water system as soon as possible, and in that way. And then as far as the public notice, that would come after the drinking water advisory is issued.
Okay, thank you. I know a lot of states have been asking that question recently about the, what does the 10-day health advisory mean? So I think that's helpful. And then, so I think this next question came in for Jennifer and Keith, and this is about the Chicago pilot study where there had been no detections of toxins in finished water. But the question is whether there were detections in raw water, and were water treatment processes the same across the study area?
Yeah, that's an excellent question, and unfortunately the study wasn't designed just for cyanotoxins, and so we don't know what the source water was like, or the detention through the treatment plants. I don't have all of the treatment trains in front of me. I'd have to talk with my colleague, Paul Bradley, but I suspect the treatments were quite different because there was multiple treatment plants involved. But regionally and within a state, you tend to have similar approved treatment types depending upon when they were built. And so if it's something that you would like USGS to get more information on, I think we could probably accommodate that.
Okay, great. Thank you. Maybe we could follow up with the person who asked the question.
Yeah.
Specifically.
The one thing I will bring up, my last understanding is UCMR4 was not going to measure the source water either. And so if that's an issue and there's an ability to adjust that with EPA, that might be something that the utilities consider in collaboration or something like that.
Now, just to go along with that, Ruth, do you require both source water sampling and finished water through the treatment train? So I imagine there might be some other states that are asking their water utilities to do that as well.
Yes, that's true. Our monitoring does focus on raw water or source water, and it's kind of interesting how we've identified what's coming into the plant. So a water system with multiple source water, say a couple different reservoirs that they could draw from. We're taking the data weekly at the plant, so we aren't necessarily able to associate it back with an individual source water body without integrating it to the data that they were pulling in at the time. But yes, it does. I think it's been a valuable tool for us to at least give warning to the water system and optimize to do both source water and finished water. And I'll just add that the only case where we do finished water only is for those purchased water systems from out of state.
Okay, great, thank you. And then regarding, this is I think for USGS again, regarding your water quality monitors being used to develop site-specific models, which parameters were being used to model the risk?
So, I mean, we're not really looking at risk, we're looking at probability of occurrence or actual concentration. And the models are site-specific, so different sites have different parameters included in the models. Some of them are based on day of year and fluorescence. Some of them include flow and oxygen, or flow and pH. But again, those are the ones that are usually most common. I think we have a couple with turbidity as well. So it really depends on the system, and we are using a variety of parameters within those. The most common are day of year and the algal fluorescence sensors.
And so to go along with that, are you using qPCR as an indicator of cyanobacteria? I know Ruth had mentioned that one, Donna Francie with USGS was using it for one of the areas. I'm not sure if you're using it in other areas throughout your collection throughout your models.
We're exploring the potential for that. The large river pilot study that we touched on is including the genetic analyses, and some of our specific system-specific studies also are using those. It depends. I know some of the studies in Ohio, some of the studies in New York, and some of the studies in Kansas right now, and some in Oregon. We have several across the country that are looking at incorporating genetics and really figuring out what those data mean and how best to interpret and use those information.
Okay, great, thank you. And then one other question, sort of looking at the models, or particularly looking at nutrient drivers. Can you share a little bit about how you're looking at phosphorus and nitrogen drivers, and then are you using those in your models, and how, and I think this actually might have been directed at Ruth in Ohio about what you're doing there, but I think USGS, Jennifer and Keith, you could chime in here too.
Yeah, USGS does a lot of nutrient work. There's a lot of status and trends and nutrient loading work done across the nation. When we're talking specifically about our cyanobacteria and cyanotoxin work, our nutrient data doesn't necessarily come into play into our models. And the types of work we're doing with nutrients are different than the models looking at probability of occurrence or concentration. Those are things that we can measure and deliver in real time. We do have some sites that have nitrate and orthophosphorus sensors, but those variables have not been significant in any of the models that we've developed.
Okay, and
yeah, I'll chime.
Ruth, yeah.
Sorry,
go ahead.
Okay. And so I'll just add that through our integrated report, we do assess the hydrological units for all of the water bodies associated with surface water, drinking water systems. And for that, we do have nitrates as an indicator. So we do look at the occurrence of high nitrates, but that's actually for its own drinking water impairment, essentially. And it would, for our inland water systems, it really is kind of up to them to look at those relationships as far as a predictive model. It's not required in rule, but I think that we are working towards more integration between the nitrate drinking water impairment and doing the surface water side of evaluating nutrient loads and performing the TMDLs, or total maximum daily loads, within a water body, as also hopefully reducing the cyanobacteria occurrence and cyanotoxin occurrence, as well as reducing nutrients. So that's where I see some integration.
Okay, great. And I think this could be a long answer for you, Ruth, but one person asked what you're doing to minimize nutrient input in the water supplies. I know you have a number of activities going on there in Ohio, so maybe you could just give us a real quick answer and maybe provide us a website to go for more information.
Yeah, I'll certainly follow up with some websites for more information. And so I think that this is more on our surface water side, where they're really going at looking at the activities in the watershed to help reduce nutrient loading, and also evaluating the flow of nutrients throughout the watershed. And in the cases where this really comes into play for the drinking water side, when you have triggered that general plan, again, only 19 of our 118 water systems have triggered that, that's a great opportunity to integrate more of the source water protection activities. And we've already seen the development of an initiative to focus on reducing nutrients in the Tappan Lake watershed. And I think that's going to be a good example of how those activities and improvements of reducing nutrient load will hopefully have the desired effect of reducing cyanotoxin occurrence. And of course, all the work for Lake Erie, which I'll refer to a separate website for that.
Great, thank you very much. I have a question here. Let's see, if a water system, hold on a minute, I just lost the question. Well, let's go on to let's go on here. Okay, here we go. If a water system only had 2 days at 10 micrograms per liter in drinking water, do you require public notice? So this is just sort of a more specific question regarding sort of your public notice protocols in Ohio there, Ruth.
Yeah, so all of the public notice protocols will be after the advisory is issued. And honestly, I'm not sure about the greater than 10. I'm assuming it means an exceedance of the action level after the 10-day window, and when does that occur? And it should really start immediately when the drinking water advisory is issued. And I'll follow up with links for our water system strategy on that, just so it's clear.
Okay, great, thank you. And then just a question for USGS on whether you're evaluating cyanotoxin blooms in Florida, in Lake Okeechobee, and they're also asking about Fort Myers and Palm Beach.
So I don't think we have any samples for this year from Lake Okeechobee, but we've worked on it in the last 2 years and working with the Army Corps of Engineers to understand the impacts of salinity on the bloom moving down versus how much water they release. And I don't think we have anything from Fort Myers or Palm Beach at this time.
Okay, thanks. And then I have another question. I'm not sure this is for Ruth or for Jennifer or Keith. Have you done any testing, sampling on groundwater wells that are hydrologically connected with surface water sources that have an active bloom, harmful algal bloom?
We've done some. We haven't done it comprehensively like we've done for surface water. We've had examples where there's some surface water exchange in ponds with groundwater, where we had detectable concentrations of toxins. And then Jennifer, your study on the Kansas River, you want to talk about the alluvium?
Yeah, there were, during a reservoir release event when waters were rising, there were cyanotoxins detected in the alluvial aquifer in the vertical collector wells that were drawing out of the alluvium. And then as floodwaters receded there, it did look like the alluvial aquifer was acting as a source for microcystin back into the river. Again, those were, I mean, the data are certainly suggestive there, but we didn't collect the data in a way that we can be conclusive about that's what was happening. But that's certainly an interesting question, and I think an area that needs more study.
Yeah, the concentrations tended to be fairly low, though.
Okay, and how about okay.
Sure. In Ohio, we had a classification of water systems that were groundwater under the influence of surface water, and they were reclassified all to be surface water systems. So we do have, I think Dayton is probably a really good example of that. So they are subject to our rules. And they, we identified those, we just have 3 of those that we identified as lower tier for monitoring. So we just looking at all the qPCR trends, and they were all entirely non-detect for microsystems throughout our kind of early study period. And so I think that's one way that we've addressed it. And then as far as any other groundwater wells, I am aware of one small set of data that microsystems were tested in groundwater wells, and I believe all the data from that was below the reporting limit, so non-detect.
Okay, great. Thank you. Well, we're a little bit past our time, so I think we'll end here. I went ahead and put our contact information up on the screen. So if people in the audience would like to contact the presenters individually, we do have the list of questions that we can follow up with some of the individual people that asked them. And just a reminder that we will have the webinar recording on ASDWA's website within the next week. And with that, I want to thank you, our presenters, first for joining us today and providing such great and interesting and useful information, and to all the audience for listening in today. And I hope you have a wonderful week and holiday. So thank you very much for joining us, and we'll end the webinar now.
Thank you.